So that immortalized cell line is one way of in vitro experiment. But there are some, in fact, many cell types may not have cell line, which means you have to use a cells coming directly from a living tissue that we call primary cell culture. And in that case, that's also in vitro experiment, but there's a little more different aspect we should consider. So first, when we call cell line, that line means those cells have been already manipulated to proliferate indefinitely and can thus be cultured for long periods of time. So from one generation to the other generation, so we call it as a passage, and it can go as many passages as possible, while primary cell culture coming directly from a living animal usually cannot proliferate indefinitely. You remember the telomere. So cells, when they divide and divide, there's only a finite number of potential to divide. So that's kind of the limitation of primary cell culture. So in fact, there's not so many cell lines. So there I put some examples of commonly used immortalized cell lines. For example, fibroblast. There's a famous 3T3 cell line. Human epithelial cell, it's a cancer cell named as a HeLa cell. Or monkey kidney cos cell, or human embryonic kidney, HEK, HEK cell. Or dog kidney epithelial cell, DOCK cell, or CHO cell, which is coming from Chinese hamster ovary. So there are like, many, but still finite number of cell lines established, and we use them for different purposes, such as when we want to study breast cancer, then there's a dozens of breast cancer cell line, which we want to use. If we want to study, let's say, cellular migration from a wound healing purpose, let's say we can use 3T3 fibroblast cell line. So this way we have well characterized those cell lines, and we can compare from one research from another researcher's results. So that serves as a good standard. So let me have a table which compares immortalized cell lines and primary cell cultures. So the advantage of immortalized cell line is it's easier to culture because those cell lines are already transformed, which means it already adapted itself into a well-controlled plastic petri dish environment. So they grow quickly, and also they are relatively homogeneous, and they're fairly well characterized, so standardized so that we can even compare with others. And also, because of this property, we can grow this cell in a quite big amount. That means we can also manipulate their genes easily. So we can create stable cell lines expressing gene of interest. So by using transgenes or overexpressing or even knock down some of the genes of our interest or not in. In comparison, primary cell culture is supposedly more relevant cell type because it's freshly taken out of the body, so it's more relevant in terms of physiology. And however, the homogeneous aspect, because primary tissue is composed of many different cell types, sometimes it's not as homogeneous when we are using primary cell culture. However, given the primary cell culture, the primary cells are from original living tissue, we can have more confidence extrapolating those results to intact living systems, such as going towards in vivo interpretation. So what are the disadvantages of primary cell culture? Because you took out from the tissue, it's not well characterized, the lifespan is not indefinite, so it has a limited lifespan, such as if you take out cells from the tissue, you may not have a long time to perform your experiment. And also heterogeneous population with high variability. So it may be more difficult to prove something with a primary cell culture. While immortalized cell lines may not have the relevant attributes or functions of normal cells because it's already quite strange cell. And another thing is, even if, let's say, the same HeLa cells, if you are growing the HeLa cell and keep growing and passing down, you know, depending on that environment, maybe 10 years ago, the published one in the literature using HeLa cell experiment may not be possible, may not be easy to reproduce. Maybe the same HeLa cell, what I'm having, may have changed their characteristics because of over too many passages. The cells may have adapted or changed its characteristics. So that's possible. So one question we can ask ourselves is, hey, we have learned about stem cells. Are stem cells closed immortalized cell lines or primary cell culture? Which is an interesting question to ask ourselves. First of all, stem cells usually are coming from an animal, so in vivo. So it's more like primary cell. On the other hand, stem cell itself has a indefinite self-renewal capability that actually kind of mimics of immortalized cell lines. So one of the proof that the cells we've got this are stem cell is whether they can like keep grow, proliferate to bring a tumor formation called teratoma. So that is one evidence, say, hey, this is a really stem cell. So in that aspect, it actually also mimics immortalized cell lines. So how to isolate cells from organisms and tissues? There are a number of ways I just want to explain with this picture. So our tissue may come from adult animal or embryo or even egg. So we put them into our petri dish and using blade to dissect them out. But still, you know, our ways of dissection might be too coarse that we could use either enzyme to digest. So using enzyme to dissect them, and then we can have a cell culture. Or very fine chopping makes still, it's kind of small explant of tissue, and that we can actually culture like a primary explant. Or further dissection, if we need, then we can have a small tissue and we call it as even organ culture. So there are various methods of cell isolation depending on whether we are having a tissue chunk or even disintegrate them into an isolated cells by use of enzymes.